Okay, morning everybody. This is far from an ideal way to explain a biology experiment to you, but we'll do our best with it. Okay, right. So one of the investigations in the sexual reproduction chapter in the flowering plant that you need to know is to use starch agar plates to show digestive activity during germination. Now, that's the first thing you need to point out if you have to answer this in the Leavenscert biology exam, is that you use starch agar. You don't use the skim milk plates, you use starch agar. And all starch agar is, it's normal agar to which a little bit of starch has been added to when you're making up the agar plates. So just to recall or recap what agar is a food source for microorganisms. So in this experiment we use starch agar to show the digestive activity during germination. And the digestive activity which you will be looking at is the digestive activity of the enzyme amylase in seeds. Okay, right, so the seeds which we will be using in today's activity will be broad bean seeds. And the broad bean seeds will contain an enzyme called amylase, which will break down the starch in the starch agar plates. Okay, so number one, if we were in the lab, the first thing I would have done for you would have been to soak 4 large broad bean seeds in water for 2 days. So the name of the seeds which we use is broad bean seeds. The reason why you soak them in water is 2 reasons. Part A is to soften the seed coat around the broad beans, also called the testa. So another name for the seed coat is the testa. So you soak them in water to soften the seed coat. And the second reason why you would soak them in water is to break dormancy. Number 2, if we were in the lab, we would use Milton disinfectant to spray down the benches, and the reason why we would do this would be to kill any bacteria or fungi on the benches. Number 3, if we were in the lab, we would take 2 of the 4 seeds and we would kill them by placing them in boiling water for 5 minutes. The reason why we want to kill the seeds is to denature the enzyme amylase in these 2 seeds. And these 2 seeds will act as the control in your investigation. Number 4, using a backed blade, you will split the 4 seeds in half. Remember, 2 of these seeds you have boiled to denature the enzyme amylase in them, and the other 2 seeds are unboiled. A safety precaution you would have here will be to cut away from your fingers to prevent cutting yourself. Number 5, then, you would sterilize all of the seeds by soaking them in Milton fluid for 10 minutes. And the reason why you would do this is to eliminate any unwanted microorganisms from the outside of the seeds. You'd wash off then the Milton fluid with water. Number 7, you're going to continue engaging in your aseptic techniques here. So you would flame a forceps by passing it through the blue flame of the Bunsen burner, and this sterilized the forceps. Number 8, you will barely open a petri dish containing the starch agar and place the unboiled half seeds face down on the starch agar in one dish, and this will be called dish A, and this will be the actual investigation. Now, before we go on to point 10 there, just to point out your aseptic techniques again that you have used here. You have flamed a forceps by passing it through the blue flame of the Bunsen burner. You only open the starch agar plates for the smallest possible distance and the smallest possible time to reduce the risk of unwanted microorganisms from the atmosphere getting into the starch agar plates. Number 10, then, you will reflame the forceps by passing it through the blue flame of the Bunsen burner, and you will place the cut boiled half seeds face down in another agar dish, and you'll label this dish B, and this is the control. Before we go on to step 11, just to note, the reason why you must place the seeds face down on the starch agar plates is to allow for the enzyme amylase to move out of the seed onto the starch agar and to digest it if it hasn't been denatured. Number 11, you place the covered dishes, both dish A and dish B, in a warm place for 2 days. Number 12, you remove the seeds from the starch agar plates and you add iodine solution to both dishes. And just recall, the test for starch is iodine. So that's why we're using iodine solution here. After 2 minutes, you pour the iodine off both of the agar plates. Okay, so let's talk about our expected results before we turn the page. So remember, dish A contained the unboiled seeds, meaning that the enzyme amylase is working in the unboiled seeds. So the enzyme amylase has moved from the seed onto the starch agar and has broken the starch down into maltose. So there's no longer any starch present underneath the seeds. So when you flood the plate with iodine, the area underneath the seeds should be clear because there's no starch left underneath the seeds. Whereas in the control dish where the seeds have been boiled, the amylase is denatured, it will not work, it will not break the starch down into maltose. Therefore, all of the plate will turn blue-black because starch is present everywhere. So let's turn the page. Okay, so my expected results are in dish A. In dish A, the agar under the seeds will stay clear according to the diagram, while the rest of the agar will turn blue-black. The reason for this is due to the digestive action of the enzyme amylase. The enzyme amylase breaks the starch down into maltose, so there's no reaction with the iodine, so the area underneath the seeds will stay clear. The rest of the plate will turn blue-black, but the area underneath the seeds will stay clear. In dish B, all of the agar will turn blue-black, and the reason for this is because amylase was denatured by boiling it. Remember, you boiled the seeds for 10 minutes at the start of the investigation. Therefore, the starch is not broken down. Therefore, all of the plate has turned blue-black. So if we just have a look at the results. So in the investigation dish A, you have your unboiled seeds. You have the clear patches underneath the seeds. There's no starch present here. The enzyme amylase has broken the starch down into maltose. Whereas in your control, in your boiled seeds, everywhere in the plate has turned blue-black. There's starch present everywhere in the plate. Okay, so that's a very quick summary of the investigation. What I would also like you to do is to go to the Senior Biology Experiment booklet, which I posted before Easter. If you scroll down to the second last investigation in the booklet, and I want you to go through the procedure and the questions relation to the experiment on this as well. I will post after this video then the investigation questions which I would like you to answer by Wednesday, please. I hope you're all keeping well and safe, that your spirits are in good form, and just to say that I'm missing you loads. Okay, thanks guys, bye.